right, drugs are drugs. So caffeine, alcohol, tobacco, these are things that can affect your physiology or how things work in your body. Chemotherapeutic is a subset of that. So it's a type of drug used to treat. So insulin will be an example. And then antimicrobials are specifically a subset of chemotherapeutic agents that are used to specifically treat infections. There is a question on these guys. So Ehrlich, he had his heart in the right place, so he came up with the idea to basically specifically kill the bacteria, but not kill yourself while you're trying to kill the bacteria, right? He didn't execute his idea that well because arsenic still can kill you too. Fleming discovered penicillin. I will never ask you dates. Domack discovered sulfanilamide. So even though it was a little bit later than penicillin, it was more kind of widely available, mass-produced. So it was kind of the first one that was used widely. Waxman found other antibiotics. So antibiotics are not man-made. They are produced by microbes against other microbes. So here you see a little penicillium or mold colony, and then staph has been streak-plated on the blood agar. There's a little zone of inhibition where there is no growth around the mold. And the reason is it's secreting penicillin, and it's killing off the staph, so it's basically got a little bit more space, a little bit more food for itself, by kind of killing the staph off, right? So, there is nothing synthetic that you've ever taken in your life. No synthetic antimicrobials or antibiotics. Maybe they were tweaked a little bit, just to increase how effective they are, make them easier to take in. But natural or semi-synthetic, nothing synthetic. So, different types of microbes produce different types of medications. Penicillin, vancomycin, rifampin, gentamicin, erythromycin. All of these are made by microbes against other microbes. We just take them and use them for ourselves. So, bacteria are good. Okay, this is something I really want you to understand. So selective toxicity. This is kind of along what Ehrlich said, right? You want to kill the bacteria, but you don't want to die with it, right? So this is possible because of differences. The more differences, the easier it is. So eukaryotic infections, remember funguses are eukaryotic. They have a nucleus, they have separate ribosomes. They're very close to us. If you've ever had a fungal infection or know of somebody that has a fungal infection, it's not a Z-pack and you're done. You have to go to the doctor. The doctor has to make sure you're healthy enough because these meds are probably going to be a little toxic to you too. And then you have to take them for a month, sometimes 2 months, sometimes even 3 months, because we are very similar. Bacteria, obviously, we're very different from them, so there's a lot more options. All right, so there's different ways to target this. So cell walls, proteins, cell membrane, a little limited because a cell membrane is a cell membrane. It's the same for bacteria, us, funguses. Everybody's got the same thing going on. Metabolic pathways, they have to be different. DNA replication, all that stuff. Oh crap, sorry. All right, so this is a big one, cell wall synthesis. So remember the brick of the wall is called peptidoglycan. It's a brick of sugar for bacteria. And I am going to come back to this or explain it in this paper, this image. All right, so this little bacteria has now infected a cut. That cut is a new home. It's going to produce these little enzymes to start eating stuff off of you, and it's going to reproduce, right? It's going to increase in numbers. Part of that includes growing in size to split into two cells, right? Binary fission. So he's getting ready, he's putting his little bricks in. There's an enzyme that comes in and glues the brick into the wall. So there's an enzyme that will have to glue it in, right? Attach it. So he keeps growing, he keeps putting in new bricks, and the enzyme starts linking it up. All right, so your beta-lactam antibiotics are characterized by this four-membered ring. Don't memorize the structures, but the four-membered ring is characteristic of all of these beta-lactam antibiotics. That's the active part of this medication. So penicillin, methicillin, right? All of these are related because of that ring. So what happens when you take these drugs is your little bacteria keeps putting in bricks, but the medication prevents the enzyme from gluing it down. So if you just stick bricks into a wall, do you think that wall's gonna work? No. So you're just sticking bricks on, the bricks are not being glued down, this wall doesn't work, and it's just like our red blood cells, they can explode due to osmotic forces. So that's how penicillin works. Alright, so derivatives of semi-synthetic, obviously to readily absorb, survive the stomach, active against more types. Vancomycin, specific for gram-positive bacteria. Bacitracin, so some of them prevent the transport, so the bricks get stuck inside the cell. Isoniazid is specifically, or FMBO are specifically for acid-fast bacteria. Alright, so obviously you have to be growing, which is usually what's happening for bacteria that are infecting you, right? It's not like they just enter and just hang out and live there. No, they're reproducing, they're eating, right? You are their new home. Okay, protein synthesis. This is another difference. Bacteria have a smaller ribosome, ours is bigger. That's a difference we can target. So, I highlighted this one also. I'm not going to ask you specifically what streptomycin, tetracycline, chloramphenicol does, but I just want you to understand the premise of how these prevent. So, bacteria have a smaller ribosome. We can use these medications to target those differences. Some will prevent them from reading the messenger RNA correctly. Some will prevent the tRNA from coming in to deliver an amino acid. Others will prevent the formation of a peptide bond. Some will prevent the subunits from coming together or from even scanning the messenger RNA. Antisense, this basically bonds to the messenger RNA that creates double-stranded messenger RNA that, once again, the ribosome can't bind to. Okay. Disruption of a cell membrane. This one's hard because we all have cell membranes and they're very similar. So the only medication that we have is amphotericin B. So this is similar to cholesterol. What happens when you stick butter out into the sun? Melts. And then when you stick that melted butter into the freezer, it hardens, right? So we have cholesterol to prevent those swings in temperature from basically melting or, solidifying our cell membrane. Funguses have ergosterol. So this is an antifungal that basically pulls that molecule together, creates a pore, and lyses the cell. So ergosterol, that's the difference. It's not cholesterol. All right. Metabolic pathways. So sulfa drugs or sulfonamides. This will be a question on the test. We do not use this pathway. We take in, so to make our nucleotides that then we use for our DNA and RNA, we take that in through our diet, so folic acid, right? So during pregnancy, you're supposed to take folic acid because your baby has lots of dividing cells, right? So you have to take extra. Bacteria, they have to use this PABA precursor, and so this is how I remember it. PABA turns into 2, so dihydrofolic acid. The 2 turns into 4, so tetrahydrofolic acid. And then a couple other enzyme steps kick in to make your nucleotides, which then are used to make DNA or RNA, which is important when you're growing and dividing, right? When you're reproducing. All right, so sulfa drugs, you can see this is that PABA part. So this is that real molecule. These are the sulfa drugs. They mimic that little shape on that precursor. So by competing for the active site with the enzyme, they basically shut down that entire pathway. This guy's busy, it's attached to the sulfa drug. Okay, so ideally, not toxic, right? To us, toxic to the pathogen. Alright, so you would still think, you're sick, let me just take something to kill everything off. No. You have your little microbiome, your little microbial community that lives on your skin, in your mouth, in your reproductive tract, in your colon, right? So doctors are often going to go, if they have time, they're going to test for what specific bacteria you have and prescribe something that's narrow spectrum. Broad spectrum is kind of the last resort. You're septic, it's in your bloodstream, it's going everywhere else. They'll put you on something broad spectrum before they figure out what it really is. The reason is because it kills off your microbiome, and then you basically have all this space, and whatever is stronger can take over. So this allows for secondary or super infections to develop from the antibiotic. Does anybody know what C. diff is? Clostridium difficile. What does Clostridium do? Does anybody know? Just based on the Clostridium name. Huh? Well, yeah, but what is Clostridium? And what is Clostridium able to do that we've learned here? Endospores. So Clostridium difficile is capable of forming endospores, and if you're on an antibiotic and you have diarrhea, they're going to test you for that. And then if you're positive for C. diff, they're going to put you on contact precautions because you take those spores from one patient, give them to everybody else. They're already struggling with whatever they're struggling with, and you just added another infection. C. diff, so you wiped out your microbiome. Have I shown you how toxic megacolon?
No, I don't think so.
So your colon gets so big, it doesn't have like the really scary ones. Hold on. They had one where somebody was laying right next to the colon, and it was like huge. Should I go on like look at Google?
Yeah.
But so essentially C. diff is because you've taken an antibiotic, the endospore is easier to last. Crap, what am I doing? I can't talk and do things. Google. Okay. But yeah, it just takes over. You get little lesions in your colon. You have some horrible diarrhea. You can spread it to people, and then, ooh, yeah, show me back. So that's just the C. diff lesions. They get pretty big, and then this is toxic megacolon. Yeah, it was worth the Google, right?
Yeah.
What? Yeah. So this person died.
Oh.
That's bad. Yeah. Well, you can actually survive without your colon. But yeah, they get so big.
That's
Yeah.
crazy.
So what is growing? Like this making grow?
This is so it's inflammation, but this is resulting from
C. diff?
yeah. So obviously that's like worst case. It doesn't happen with everybody, but so we'll talk more about C. diff later on. There's some pretty nasty stuff we can talk about. Okay. All right. So you're often right broad spectrum, last resort, right? Your microbiome is good for you. They produce vitamins for you in your colon, right? There's only limited space, limited resources in there. So, you know, like whatever's coming in pathogenic wise has to compete with your already established microbiome. Okay, this Kirby Bauer we are going to do next week. So we'll talk about it next week. Okay, good. So once again, disruption of your normal microbiome can result in secondary infections. So yeast infections, when you're on a bacterial antibiotic, yeast is a eukaryotic fungus, right? So antibiotics are not gonna, yeah, so that's why you can have a yeast infection due to an antibiotic. Okay, resistance. So you are taking an antibiotic and you feel good by day 3 and you're just like, screw it, I'm not gonna finish it. Well, you've now exposed these bacteria to that drug, and they are exposed, they know about it, and that facilitates the growth of resistant strains. So always finish your antibiotics. I know I'm guilty of it because we would go to Mexico and get them back to like a little bottle. Don't do that. Yeah, because you basically foster more resistance. So resistance factors are, this is an example, beta-lactamase. It's an enzyme that, so MRSA stands for methicillin-resistant Staph aureus. This guy produces this enzyme beta-lactamase that clips the active ring of that chemical, and now penicillin doesn't work. So if you're resistant to penicillin, you're resistant to methicillin, you're resistant to anything that has that 4-membered ring. So likely none of that class of antibiotics you're able to use. The scary part of this is that conjugation, you don't even have to be the same species. You can just share that with whoever is next to you. Just like, hey, you want this resistance factor? And they can just give it to each other. So our plasmids exchanged. Okay. All right, this is a really interesting documentary. It's talking about how we're running out of stuff. Resistance is like super high. There's a lot of resistant strains, and where like, you know, you just get a cut, you're fine, you take some medication, there's I think 2 people, 3 people. So there's one little girl who got like she caught MRSA from like picking a scab, and then she had this crazy infection. They couldn't have, it was resistant, so no medication that they could use to treat her. It was spread to her lungs. They had to it was horrible. She survived, but then later on, after the documentary was over, she died. But she survived, but she wasn't all there. Like it was, yeah. That infection kind of messed her up. The other one was this guy who was doing I don't know, mission work, and he ended up falling, being run over by a train, and his leg got cut off, and he had an infection, and it kept spreading up and up, so they had to keep cutting off more and more of his leg. So when they finally flew him back home, they had no antibiotics to treat him. It was all resistant strains. So they had to go back to toxic forms of antibiotics that were actually starting to kill him too. He survived. He was there, but the girl, she did survive, but then she didn't. So they even talked to executives because they're like, well, it's very expensive to come up with these medications. Why would you spend billions of dollars for something that somebody's going to take in 4 days? It's not like blood pressure where you have to take it for the rest of your life, or diabetes meds where I've got you forever. This is a Z-Pack and you're done. Why would I spend billions of dollars? So there's no money in it. That's why people aren't looking for more. So I would watch it. It's pretty